Hello, Biology 202 students. Welcome to your online lecture. The digestive system is a remarkable and complex network that transforms the food we eat into essential nutrients and energy for our bodies. Spanning about 30 feet from mouth to anus, this system efficiently processes and absorbs the nutrients we need while eliminating waste. As a fun fact, the small intestine alone is about 22 feet long, and the surface area of the entire digestive tract is roughly the size of a tennis court. This is to maximize nutrient absorption. How exciting! Today we will cover the key components of the digestive system. We'll start with the functions of the system and then move on to the alimentary canal, which includes the mouth, teeth, esophagus, stomach, small intestines, large intestines, and anal canal. We will discuss the histology of the stomach and small intestine in detail. Next, we'll explore the accessory organs, including the salivary glands, pancreas, liver, gallbladder, and the greater and lesser omentum. We will then delve into the liver and its hepatic triad, including a look at its histology. This will be an exciting lecture, I hope. So make sure to grab a snack and a beverage so you can do some digesting as you learn about digesting and this incredible system. Let's introduce the functions of the digestive system. First, we have our favorite, ingestion of food. This involves taking food into the mouth. The food then undergoes chemical and mechanical breakdown, where it is physically and chemically broken down into smaller components. The next function is storage of food. So our whole digestive system provides an area for our food to rest while we digest it. Next, we have absorption of nutrients and water. This occurs as the system absorbs necessary nutrients and water from the food. Additionally, the digestive system is involved in the synthesis of vitamins, producing certain vitamins necessary for our bodily functions. Finally, the system handles the elimination of waste, getting rid of the undigested and unneeded parts of food. The digestive system is composed of two main parts, the alimentary canal and the accessory organs. The alimentary canal, also known as the gastrointestinal tract or the GI tract, is a continuous tube that extends from the mouth all the way to the anus, encompassing the mouth, pharynx, esophagus, stomach, small intestines, large intestine, rectum, and anus. This channel is responsible for the direct passage and processing of food. In addition, the accessory organs, let's just move this over, including the teeth, salivary glands, pancreas, liver, gallbladder, vermiform appendix, and the greater and lesser omentum, which all play a crucial role in digestion by producing and secreting enzymes, bile, and other substances that aid in the breakdown and absorption of nutrients. Together, these components work to ensure the efficient digestion, absorption, and elimination of food and waste products. So let's break into the details. Within the alimentary canal, there are three layers that are seen again and again and again, each with specific functions. The innermost layer, the mucosa, is lined with epithelial tissue and is responsible for the secretion of mucus, digestive enzymes, as well as the absorption of nutrients. Beneath the mucosa lies the submucosa, a connective tissue layer that contains blood vessels, nerves, and glands, providing support and nourishment to the surrounding tissues. Then there is the muscularis externa, This consists of two layers of smooth muscle, an inner circular layer and an outer longitudinal layer, which work together to produce peristaltic movements that propel food through the digestive tract. The outermost layer is the serosa. It is a protective connective tissue covering that reduces friction between the digestive organs and the surrounding structures. It also holds everything together. The mouth is the very beginning of the alimentary canal. This is also the very first place we see mechanical and chemical breakdown of food. The teeth, especially the molars, play a critical role in mechanical digestion by grinding food into smaller pieces, increasing its surface area for enzymes to act upon. The salivary glands, which are also the very first accessory organs we see, include the parotid, sublingual, and submandibular glands. These secrete saliva, which contains enzymes like amylase that begin the chemical breakdown of carbohydrates. Saliva also lubricates food, forming it into a bolus. The tongue then aids in manipulating and guiding this bolus towards the pharynx and into the esophagus. The esophagus is a part of the alimentary canal, and it connects the pharynx and the stomach. It serves as a conduit for that bolus of food. It features an upper section composed of skeletal muscle to allow voluntary control of swallowing, and it transitions to smooth muscle, which facilitates the involuntary peristalsis to propel the bolus towards the stomach. Lined with stratified squamous epithelium, the esophagus is protected against abrasions from food. At the lower end, the esophageal sphincter prevents acidic stomach contents from refluxing out, ensuring one-way movement of ingested material into the stomachs for further digestion. So here on the left is a great GIF of peristaltic movement of a bolus from the esophagus down to the stomach. And on the right here, we have the histology of the esophageal stomach junction. So here on the left, you can see the stratified epithelium here, and then on the right, you can see some gastric pits, and we will talk a little bit about those in the next slides. The stomach is a key organ in the digestive system, receiving food from the esophagus through the lower esophageal sphincter into its lumen. As part of the alimentary canal, the stomach consists of the same four layers: the mucosa, the submucosa, the muscularis, and the serosa. Students should consider these layers as we discuss the stomach structure. Food enters the stomach at the gastroesophageal junction, up here, and moving through the cardia, right here, then to the fundus, over here, and into the main part, the body of the stomach. The stomach has a greater and lesser curvature, and its muscularis externa has three layers: the longitudinal, circular, and oblique, which facilitate thorough mixing and churning of food for a continuous mechanical breakdown of the food. There's also the rugae, or folds, allowing the stomach to expand significantly. Food exits the stomach through the pyloric sphincter into the small intestine. And before it does exit, it actually goes through a name change. So we had a bolus of food coming through here, and then our stomach added its fluids and started churning it up, and now it's called chyme. Within the stomach's lining, gastric pits lead to gastric glands that contain cells crucial for digestion. We have parietal cells, which produce hydrochloric acid, which aids in the breakdown of food and creating a hostile environment for pathogens. Then we have chief cells, which secrete pepsinogen, which is converted to pepsin in the presence of hydrochloric acid to digest proteins. A common pathology associated with the gastroesophageal junction is gastroesophageal reflux disease, or GERD. GERD occurs when the lower esophageal sphincter fails to close properly, allowing acid from the stomach to come and reflux up into the esophagus. This can cause some symptoms like heartburn, chest pain, and regurgitation. and can cause more serious complications like Barrett's esophagus if left untreated. As we start looking at some histology of the digestive system, I'd like to introduce a very helpful tool. This is called a diatomaceous key, and it's for the digestive system. So it's kind of like a choose your own adventure. So we start off here at 1, and then we read gastric pits in the mucosa. If there are gastric pits in the mucosa, then we know we are in the stomach. If there are no gastric pits, then you proceed to 2. So here is 2. Are there villi in the mucosa? If there are, proceed to 3. If there are no villi, proceed to 4. So let's take a look at an image here. Let's start off. Are there gastric pits? Yes, you should recognize this histology image, and there are gastric pits right here. So we're looking at the stomach over here. After the chyme leaves the stomach through the pyloric sphincter, right here, it enters the first section of the small intestine. The small intestine has 3 sections: the duodenum, the jejunum, and the ileum, each with unique structures and functions. All sections contain the same 3 layers found throughout the alimentary canal: the mucosa, the submucosa, and the muscularis. The most crucial layer for nutrient absorption is the mucosa, which has villi, tiny projections that significantly increase the surface area for absorption. So this is what you need to remember for the small intestines. Take a look at the mucosa, look for villi, and then take a look at the submucosa, because there might be something interesting there, depending on which section you're looking at. Zooming in on the microvilli, we see the mucosal layer, containing blood vessels and lymphatic vessels called lacteals. The lacteals absorb fat while the blood vessels absorb nutrients. And this includes some of the chemicals from prescription medications, coffee, or like sugar. These nutrients are often then transported through the bloodstream to the liver for processing. Now let's discuss the unique features and functions of the duodenum, the jejunum, and the ileum. So the duodenum is the first segment. It's about 10 inches long, and it receives chyme from the stomach, along with bile from the gallbladder and digestive enzymes and bicarbonate from the pancreas. It contains duodenal glands, also known as Brunner's glands, that secrete an alkaline mucus to help neutralize the acidic chyme. So we have the gallbladder here, and we have the cystic duct connecting it to the bile duct right here. And so this is where bile is secreted into the duodenum down here through the hepatopancreatic duct. And this combines both the bile duct and the pancreatic duct. If we move up here, we have the common hepatic duct, which leads into the liver. Next, we have the jejunum, which is approximately 6 feet long. It is the primary site for nutrient absorption and water absorption. It is characterized by prominent villi and microvilli, which increase the surface area for absorption. The jejunum lacks glands in the submucosa, focusing instead on absorbing nutrients through its highly vascularized mucosa. The last section of the small intestines is the ileum, which is about 10 feet long and continues the absorption process. It also contains Peyer's patches, which offer immune support to the digestive tract. This part of the intestine connects to the large intestine at the ileocecal junction, regulating the flow of digested material into the colon, right here. Alright, remember this diatomaceous key here? Well, let's use it very quickly to go over the histology of the small intestine. So here are our 3 options: a submucosa with duodenal glands, a submucosa with nothing, it's boring, and a submucosa with lymph nodules. So let's take a look at these 3 images here. Let's see if you can find the muscularis, submucosa, and mucosa. So what was the defining characteristic of all the small intestine? We have villi in the mucosa. So here are villi. Here, here, and here. They're all little finger-like projections into the lumen of the intestine. So the next tissue right below that is the submucosa. So you can see here, here, and here we have the submucosa. Something wild's going on in this one. And then just below that we have the muscularis, the smooth muscle that's peristaltically moving this bit of chyme. So let's start on the left here. Notice anything? Well, if you look closely, you can see some spottiness within the submucosa. Right here, and those are glands. Do you remember the glands that are in the small intestine? We have duodenal glands. So this is the duodenum. Let's go to the middle one here. Do you see anything going on in the submucosa? I don't see much going on here. I see some blood vessels, but nothing really interesting. So this is boring, which means it's probably the jejunum. Now, the far right, I know you notice what I'm noticing. These dark patches right here, they're large, and these are the Peyer's patches. These are the lymphatic tissue found in the ileum. So this is the ileum. The pancreas is a vital organ with both exocrine and endocrine functions. These play roles in the digestion and blood glucose regulation. The exocrine function of the pancreas involves the production and secretion of digestive enzymes and bicarbonate into the small intestine. The acinar cells of the pancreas produce enzymes such as amylase, lipase, and protease, which are released into the pancreatic duct and then into the duodenum. These enzymes help break down carbohydrates, fats, and proteins in the chyme, facilitating nutrient absorption. Additionally, the pancreas secretes bicarbonate, which neutralizes the acidic chyme from the stomach, creating an optimal pH for enzymatic activity in the small intestine. Next, we'll talk about how the pancreas manages blood glucose with glucagon and insulin, otherwise known as the endocrine functions of the pancreas. The endocrine functions of the pancreas are crucial for blood glucose regulation. The pancreas contains clusters of cells called the islets of Langerhans, which include alpha and beta cells. The alpha cells secrete glucagon, which raises blood glucose levels by stimulating glycogenolysis and gluconeogenesis in the liver. It's extremely important to understand the difference between glycogenesis, gluconeogenesis, and glycogenolysis. Glucogenesis is the process of creating glycogen and depositing it into the muscles and liver. Glycogenolysis is the process of breaking down those glycogen storages so you can have glucose in your blood. Gluconeogenesis is the production of glucose with the use of amino acids and lactate and glycerol as precursors from non-carbohydrates. So it's making glucose from something that wasn't glucose. So again, we have alpha cells that secrete glucagon, which raises blood glucose levels by stimulating glycogenolysis and gluconeogenesis in the liver. Conversely, the beta cells produce insulin, which lowers blood glucose levels by promoting the uptake of glucose into cells and inhibiting glycogenolysis and gluconeogenesis. So take a look at this depiction of the management of normal blood glucose levels. When we have a nice big sugary meal, we have higher glucose levels. Then our pancreas releases insulin from our beta cells in our islets of Langerhans. This insulin goes to the liver and our cells saying, let's take up that glucose in the blood, let's stimulate the production of glycogenesis, which puts glycogen into the liver and muscles, and our cells get that glucose that's just in our blood. Then our blood glucose levels go down, which tells our pancreas to release glucagon, which will stimulate glycogenolysis, which breaks down those glycogen deposits we made, and gluconeogenesis, which promotes the synthesis of glucose in our tissues. Then our blood levels go back to normal. So that's the cycle. Proper functioning of these endocrine cells ensures balanced blood glucose levels, which is vital for overall metabolic health. Dysregulation of these processes can lead to conditions such as diabetes mellitus, where insulin production or action is impaired. Diabetes mellitus is classified primarily into two types, type 1 and type 2. Type 1 diabetes is an autoimmune condition where the immune system attacks and destroys the beta cells in the pancreas, leading to little or no insulin production. This type of diabetes often manifests early in life and requires regular insulin administration for management. Type 2 diabetes, on the other hand, is characterized by insulin resistance, where the body's cells do not respond effectively to insulin. Initially, the pancreas compensates by producing more insulin, but over time it cannot maintain significant insulin production. Type 2 diabetes is often associated with lifestyle factors such as obesity and inactivity, and can sometimes be managed with lifestyle changes, oral medication, and insulin therapy. Both types of diabetes require careful monitoring and management to maintain blood glucose levels within a healthy range and prevent complications. So here's the histology of the pancreas. You can see acinar cells pretty much everywhere. They're the dark purple ones. And then you can see islets of Langerhans or pancreatic islets in these little spotted areas. They're a little lighter staining and they make little spots. So our exocrine function is all the purple cells around here, and those secrete our digestive enzymes and sodium bicarbonate. And the islets secrete our insulin and glucagon. The liver is a multifunctional organ that plays a pivotal role in digestion, metabolism, and detoxification. One of its primary functions is the production of bile, which is essential for the emulsification and digestion of fats in the small intestine. The liver processes nutrients absorbed from the digestive tract, converting glucose to glycogen for storage and regulating blood glucose levels. It also metabolizes proteins and fats, producing essential proteins which make up blood plasma like albumin and clotting factors. Anatomically, the liver is divided into 4 lobes: the right, left, quadrate, and caudate lobes. Blood from the digestive tract goes into the portal vein here and goes into the liver, and then it leaves the liver from the hepatic veins. The liver's detoxification functions include breaking down and eliminating toxins, drugs, and metabolic waste products from the blood. Blood from the digestive organs passes through the hepatic portal vein into the liver, where it's filtered through a network of sinusoids. Hepatocytes, the liver cells, process and detoxify this blood, producing bile from bilirubin, which is waste from the dead red blood cells, and bile salts that is stored in the gallbladder until needed for digestion. The gallbladder is in charge of storing and concentrating this bile, so it can be used whenever you eat a fatty meal. Bile is amphipathic, meaning it has a hydrophilic side and a hydrophobic side, which is good because that is what makes it good at emulsifying fats. This is considered mechanical digestion because emulsifying fats is literally breaking fat into smaller and smaller micelles or chylomicrons, which you learned about in the lymph lecture. Within the liver, hexagonal lobules consist of hepatocytes arranged around a central vein, with the hepatic triad at each corner containing a branch of the hepatic artery, a branch of the hepatic portal vein, and a bile ductule. Blood from the hepatic artery supplies oxygen, while blood from the portal vein supplies nutrients. The bile produced by hepatocytes drains into the bile ductules and eventually into the common bile duct, which leads to the gallbladder and the small intestine. So here on the right is a histology image of the liver. You can see here a lobule. There's usually a dark outer border, and there are tons of hepatocytes within here. And then there's a central vein right here, which will go into the hepatic vein and eventually lead into the inferior vena cava. Then we have the hepatic triad, which includes a branch of the hepatic artery, a branch of the hepatic vein, and a bile duct. We know how to identify veins and arteries. Arteries have thicker walls, and veins have a larger lumen. And bile ducts have a darker staining endothelium. So if you look at this corner of the hepatic lobule, you'll see here a vein. Here is a artery. And then right here is a hepatic bile duct. Gallstones are hardened deposits that can form in the gallbladder, typically composed of cholesterol or bilirubin. These stones can vary in size and may cause blockages in the bile duct, leading to intense pain. If a gallstone obstructs the cystic duct or bile duct, it can result in inflammation of the gallbladder, or cholecystitis. Treatment for symptomatic gallstones often involves surgical removal of the gallbladder, known as a cholecystectomy. The large intestine is the final part of the digestive tract, tasked with absorbing water and electrolytes, forming feces, and ultimately expelling waste from the body. It's about 5 feet long, and it includes several regions: the cecum, vermiform appendix, ascending colon, transverse colon, descending colon, and sigmoid colon, and then rectum. Unlike the small intestine, the large intestine has a wider diameter and features haustra, pouch-like segments that give it a segmental appearance. Its inner lining, or mucosa, lacks villi, but it's rich in goblet cells, which secrete mucus to ease the passage of feces. The rectum then stores feces until defecation. The vermiform appendix is a small tube-like structure attached to the cecum, located at the junction of the small and large intestines. The appendix is about 3 to 4 inches long and has a narrow, worm-like shape, hence the name vermiform, which means worm-shaped. While its exact function is not fully understood, it is believed to play a role in the immune system, particularly in harboring beneficial gut bacteria. Despite its relatively minor role, the appendix can cause significant issues if it becomes inflamed, leading to a condition known as appendicitis, which often requires surgical removal. The anal canal, the final segment of the large intestine, is approximately 1.5 inches long and is responsible for the final elimination of waste. It is lined with stratified squamous epithelium to protect against abrasions as feces passes through. The canal has 2 sphincters: the internal anal sphincter, made of smooth muscle and under involuntary control, and the external anal sphincter, made of skeletal muscle and under voluntary control. These sphincters work together to regulate the passage of feces, ensuring controlled defecation. The internal sphincter even helps distinguish whether you have to pass gas or defecate. So again, we'll look at our diatomaceous key. Definitely study this thing, because it will help you out. Alright, look at our image here. We see a ton of goblet cells. So this is our large intestine. The greater and lesser omenta are folds of the peritoneum, a membrane that lines the abdominal cavity. The greater omentum is a large apron-like fold that hangs down from the stomach and covers the intestines. It contains fat deposits, blood vessels, and immune cells, helping to cushion and protect the abdominal organs, as well as playing a role in immune response. The lesser omentum is a smaller fold that connects the stomach and the beginning of the small intestines, the duodenum, to the liver, providing a pathway for blood vessels, nerves, and other structures to reach the liver. Together, these structures help support the organs and protect them within the abdomen. Congratulations, you made it to the end of the lecture. Thanks for your attention, and make sure you check the syllabus for any upcoming due dates. Have a great rest of your day.